Great.
Okay, welcome everybody to this Validate seminar, the first in a series focusing on vaccines for skin and neglected tropical diseases. A little bit of a brief background. In May this year, Rachel and I and a couple of people in this currently in the Zoom were in Bogota for a workshop focusing on vaccines for skin NTDs. We brought together around 28 experts from, I think it was 11 countries, if I remember correctly, where we discussed challenges, explored collaboration solutions, and really focused on the ideas around developing vaccines for, more specifically, Leishmaniasis, Buruli ulcer, leprosy, and cutaneous tuberculosis. The talks were so good, including the two you're going to hear today, we thought we'd share it with the wider Validate community. And the two talks we have today were from the pathogenesis section of the workshop. We're going to start with Dr. Fernanda Novais, who is the assistant professor at The Ohio State University and the co-director of the Host Defense and Microbiology program at the Infectious Diseases Institute. Fernanda will be exploring immune responses to Leishmaniasis and her lab's investigations into immune mechanisms that promote tissue damage without contributing to parasite control. And Professor Rachel Simmons will follow. And Rachel is a professor of Immunopathogenesis at the, Immunopathogenesis at the University of Surrey, and studies the mechanisms and actions of Mycolactone, the toxin of Buruli ulcer. Today, Rachel will explore Mycolactone and how it disrupts cellular process in Buruli ulcer. So first of all, I'm going to hand over to Fernanda. Are you ready to go?
Ready. Let me just share my screen.
And I'm going to turn my camera.
Can you see that?
I can see that perfectly.
Okay. Awesome.
Message in the chat if people can't see.
Okay. Well, thank you very much to the Validate community for inviting me here to talk about the work from my laboratory. We study cutaneous leishmaniasis, which is a neglected tropical disease of the skin. And in my lab, we're particularly interested in the immune responses related to this pathogen. So I like to start with this because it really highlights that the cutaneous form of the disease and the mucocutaneous form of the disease are really not one disease, but a spectrum. And you can have individuals with cutaneous leishmaniasis with this diffuse form of cutaneous leishmaniasis, in which the disease is primarily caused by a high pathogen load. So if you biopsy one of those lesions from these patients, parasites are very difficult to find. And what you do see, parasites are very abundant, actually. So what you do see is that you have a lot of macrophages that are highly infected with Leishmania parasites. And what you don't see is a lot of T cells, which really argues for that the presence of T cells is really critical for the control of the parasite. And as you migrate from this end of the spectrum to this end of the spectrum, and I'm going to include both the individuals with severe form of the disease at the site of infection, which is the skin, and also those individuals in which the parasite metastasized from the skin to the nasopharyngeal mucosa. And in those individuals, pathogens are difficult to find. But what you do see is a huge influx of T cells. So it really argues, and this is not unique to Leishmaniasis, that you can have in one disease the severity caused by an uncontrolled pathogen growth or an efficient control of the pathogen, but immune-mediated pathology. And in my lab, we're really interested in what causes this, because it's really unfortunate for these patients that there are no vaccines that are approved for human use, and treatments that are currently used are often ineffective. So, for example, the patient population that I work with are infected with Leishmania brasiliensis parasites in Brazil, and they're treated with this standard therapy, which is called pentavalent antimony, which is a heavy metal that requires multiple days of injections. Essentially, half of these patients will fail the first round of treatment, and they have to go through a second and sometimes a third round of treatment. And we think the treatment is so ineffective in these patients because it targets the parasites. And as I showed you, these patients don't have as much trouble controlling the pathogen as they do controlling the immune response. So the work in my lab is really trying to understand the immune responses that are protective, that we want to encourage for vaccine development, but also those that are associated with more severe disease and immunopathology. So if we understand what pathways are involved in this process, then we can think of either dampening them when we're disconsidering them for vaccine development, but also thinking about them as to be targeted by new treatments. So Leishmania is delivered to the skin by the bite of an infected female sandfly, and to control the disease, it's very well described that you require dendritic cells, particularly producing IL-12, that leads to the differentiation of CD4 T cells into Th1 cells that produce interferon gamma, and these interferon gamma producing cells then activate the infected macrophages to control Leishmania. So this is the type of response that you want to encourage in vaccine development, that you want to not target when you're thinking about new immunotherapies for patients. However, a lot of work that I did as a postdoc and now in my lab, and also by many other groups, what we found is that there is a subset of T cells called CD8 T cells, or cytotoxic T lymphocytes. And these cells, their primary function in the skin seems to be to be cytotoxic. These cells produce very little to no granzyme, interferon gamma, which is the protective cytokine. But what they do is produce perforin granzymes. Perforin opens a pore on a target cell, delivers granzymes inside the cell, and that kills that infected cell. And although this is very counterintuitive, the killing of the infected cell does not lead to the control of Leishmania. In fact, we see parasites being released healthily, and then they infect new cells. And these parasites can also metastasize. And in the mouse, we see the development of multiple lesions throughout the body. In patients, we hypothesize this is involved in the development of mucosal disease after the primary skin infection. And this overwhelming cell death that CD8 T cells cause activate inflammatory responses through this inflammasome called the NLRP3 that leads to the processing of this very inflammatory cytokine L1 beta that ultimately leads to chronic neutrophil recruitment, severe inflammation, and the chronic ulcer that we see in patients. And we know this is a critical pathway in both mouse models and in patients because we've done a lot of work in both mice and patients. But also, if we use drugs that either target and dampen cytolytic responses by CD8 T cells, or block the inflammasome, or block IL-1 itself, we can block the disease that's mediated by CD8 T cells. So this is an example of a mouse untreated or treated with one of these drugs, in this case, anakinra. And what is curious, and I'll go back to that at the end, is that there's no better control of the parasites. In these mice, the control is just as bad. What we don't see is immune-mediated pathology. So as I was trying to migrate from to my independent position, I started my lab in August 2020. We were trying to consider, what were the questions that were open in this model as a way of starting my lab with something that I was very familiar with. So there are many open questions that we're trying to answer, but the one that I want to talk about today is this one. So it was clear to us how CD8 T cells promoted disease. What wasn't understood at the time was why. So why are CD8 T cells pathogenic, and why aren't they protective in this disease? And if we understand this, we can think about other strategies, right, to dampen CD8 T cells upstream of cytolytic program. So the reason I decided to start answering this question is because there was a dual role of CD8 T cells in cutaneous leishmaniasis based on more older literature, and then that contrasted with what I was studying. So there was data from the past showing that CD8 T cells can produce interferon gamma and be protective. But if we try to make sense of what really they showed, is that CD8 T cells very early upon infection, within the first few days, they make a lot of interferon gamma. And this interferon gamma they produce early on really pushes these dendritic cells to make IL-12 and then produce interferon gamma and control the parasite. However, once they get to the skin, and that's what we were studying, right? CD8 T cells in lesions from patients and mice are primarily cytotoxic. They don't produce as much interferon gamma. They primarily focus on perforin granzymes, resulting in immunopathology. So with that paradox, what we hypothesize is that the function CD8 T cells exhibit in this disease depends on the tissue they're located. So while CD8 T cells in draining lymph nodes are protective because these responses and the generation of T helper 1 cells happens in draining lymph nodes, once they get to the lesion, these CD8 T cells change their behavior and they become pathogenic. So I will not talk about this today. I'm primarily going to focus on the mechanisms of induction of the cytolytic program and the negative outcomes it has in CD8 T cells. And all the work that I'm going to present today, it has been done by Erin Fowler, which is a graduate student in my lab. So the first thing that Erin did to test this hypothesis is to really check whether the differential expressions of granzyme B between draining lymph nodes of infected mice and lesions. So there are a couple housekeeping things I'd like to say. Parasites are found in draining lymph nodes and lesions. And all the slides that I'm going to show you is going to have how we've done the experiment. If there's a flow cytometry flow plot, I'm going to have how we got to this population here. So this is the pre-gating strategy, representative flow plots, and a graph from one experiment that has been reproduced many, many times. So, and this graph here, as you can appreciate, if we look in draining lymph nodes, CD8 T cells produce less than 1% of granzyme B. This is actually the most we've ever seen, these little dots here. But if you look at the lesion, depending on the experiment, there's anything between 40 to 80% of the CD8 T cells that are expressing granzyme B. And I'm using granzyme B as a marker for the ability to be cytotoxic and pathogenic. So this is true for infections with Leishmaniasis and Leishmania major, for example. So this led us to hypothesize that there's something that happens when they leave the lymph nodes that they can only find when they get to the skin that then changes the CD8 T cell behavior. And then to test this, we infected mice that either express CD45.1 or 2. So we know these are the donors and these are the recipient cells. Both groups are infected. And then we can take the granzyme B negative cells and put them directly into the skin and see, is that sufficient to change how these cells behave, right? Very early, at 36 to 48 hours post-infection. And when we did this, what we found is that indeed, if you just expose this previously granzyme B negative cells to the lesion of the skin of an infected mouse, that's sufficient to change their behavior and they begin expressing granzyme B. While if these cells migrate back to draining lymph nodes, then they remain granzyme B negative. So this is really telling us that there's something happening in the skin that changes how these cells behave. Right, so our hypothesis is that there's a signal that changes the transcriptional profile of CD8 T cells, maybe 1 or 2 transcriptional factors or many, that induces the expression of preferring granzymes in these cells. And to try to understand what this signal could be, we decided to purify CD8 T cells from draining lymph nodes and lesions of infected mice, and then do transcription analysis in these cells. And then beyond things that we would expect, the cytolytic CD8 T cells are only lesions compared to draining lymph nodes. One of the things that was the most striking and different between these two subpopulations was that if you look at by pathway analysis, what are the pathways that are differently CD8 T cells and lesions are responding to? Hypoxia-related pathways, and we have 5 of them shown here, were only found in CD8 T cells in the lesions. So hypoxia is not something I've studied before I encountered this piece of data, so I wanted to make sure there was any relevance to human disease. Because we have banked multiple transcription analysis from patients, I decided to look at these pathways now in our patient population, comparing healthy skin and Virulence CL lesions. And when we do this, while in the blood, if we compare transcription, the profiles of healthy subjects and CL patients, there's no difference in the blood. We see that there's a significant induction of hypoxia, one of the hypoxia pathways here, but this is true for all of them, and I'll show that later. But that's induced only CL patients compared to healthy subjects. And I also want to point out that there's a huge variability in patients, right? Healthy subjects also have a good spread. So I'm going to go back to this variability and how we use that to inform how this is all working. So to summarize, it seems like if you purified CD8 T cells in lesions, they're responding to hypoxia. And if we look at the entire lesion in patients, there is an induction of a hypoxic signature in patients. So is this important, right? What does that mean? So hypoxia is something that happens when a cell, the oxygen supply does not meet the demand, and cells are exposed to different oxygen levels as they're navigating through our body. And hypoxia is very widely studied in cancer biology because it has a great impact in tumor cells themselves. So depending on the tumor, but the majority of tumors, the more hypoxic they are, the more aggressive and more difficult to treat they are, because tumor cells are more invasive and metastatic in the hypoxic conditions. They have a defect in apoptosis. So this is widely studied. But if you look at immune cells, each immune cell subset will have a different response to hypoxia. This is very cell type dependent. But for CD8 T cells, the consensus in the literature, I looked back, is that if you culture CD8 T cells in the low oxygen conditions, they express more granzyme B, and they're better able to kill a tumor cell compared to CD8 T cells that are cultured in our tissue culture room, which has around 20 to 21% of oxygen. So this suggests to us that maybe it is this low oxygen level that is responsible for changing how CD8 T cells behave. But then we wanted to make sure that there are indeed different oxygen levels that CD8 T cells are exposed to in these two tissues. So we use Oxy4G4, which is a phosphorescent probe that's quenched by oxygen, to infer the amount of oxygen within the tissue. And what we found is that, yes, CD8 T cells in adrenal lymph nodes are exposed to X amount of oxygen, and when they get to the lesions, they're exposed to extremely low levels of oxygen. If we look at a tissue as a total, right? So if we change minimums of mercury, there is some mice had less than 0.1% oxygen. So these lesions are extremely low in oxygen. So is that sufficient then to change these cells to become cytotoxic? Is it enough to just take these cells, put them in hypoxia, and will they become then cytolytic? And this is important because there are other signals in the skin that could be cooperating in this process. So when we did this, and we took cells from draining lymph nodes of an infected mouse and then cultured them in 20% oxygen, or 1% in this case, what we see is that there is a significant increase in the ability of these cells to express granzyme B, and this is associated with the cytolytic program of CD8 T cells. So to summarize a lot of published data that you can find in these two papers, what we found is that, yes, when CD8 T cells get to the lesions, they're exposed to low oxygen levels. This triggers the induction of this one transcription factor called BLIMP1 that triggers then granzyme B expression in CD8 T cells. In contrast, hypoxia also induces the production of perforin, which is required for granzymes to get in, but this is independent of the expression of BLIMP1 and may involve another transcription factor, and we're trying to understand how this is working. So ultimately, what hypoxia does, at least in CD8 T cells, is to produce a great cytolytic program leading to the development of chronic ulcer and more severe disease in the mouse, and we think this is also true in patients. So in the interest of time, I'm not going to show you all the data, but I think there's something that may be translatable to other infectious diseases, particularly of the ones of the skin, that which is why is these lesions hypoxic in the first place, right? Why are they so low in oxygen compared to normal skin or to the adrenal lymph nodes? So to try to understand this question, we went back to our transcription analysis in patients, and I showed you that there's this huge variability between patients, right? So you have individuals in yellow that are more similar to a healthy individual skin. There is a group of individuals here, a big group, that has this intermediate phenotype, and then you have these individuals with more hypoxic lesions. We didn't make these divisions by eye. We actually performed an unsupervised hierarchical clustering using all the hypoxia-related pathways. And then you can see in gray, you have the healthy subjects, and in purple, you have the CL lesions. And then you have these we call baseline because they group more like baseline healthy skin. Then you have these patients with intermediate phenotype and these more hypoxic cells. Tissues. So we wanted to ask, what is different between these two groups, right, that may be indicating why are these lesions more hypoxic? And when we did this, and we performed a bunch of transcription, different analysis in this transcription analysis, but one thing that stood out is that the lesions that are more hypoxic have a granulocyte signature, meaning in those lesions, there's a lot more neutrophils. And we performed MCP counter analysis, which shows that there are more neutrophils in number in those lesions that are more hypoxic compared to those lesions that are more baseline. So what is nice about this is that this generated this hypothesis that we can go back to in the mouse and ask, is this truly associated with more severe disease, right? Is neutrophils associated with the hypoxia that we see in our mouse models as well? And to test this, we decided to use this this compound called pimonidazole. So pimonidazole, you can inject in the mouse, it will go inside cells, and it's only going to bind covalently in cells that are experiencing low oxygen, less than 1%. And when we do this, you can see that there are regions. So this is a skin, infected skin, right? So you have the epidermis, you have the dermis. This is black areas, the cartilage. And then you have areas, and I'm going to highlight this, right? Where there's low pimonidazole staining surrounded by areas with intense staining. This is true here as well. And then these areas here have low pimonidazole compared to others. So again, we decided to look at this variability and try to understand what that means. And if we take this area here, for example, and we look at the intensity of pimonidazole in this area, as you can appreciate here, you have this low hypoxic cells areas compared to high hypoxic in these valleys here. So if we try to overlay where the neutrophils are in relationship to this pimonidazole staining, what we find is that really there's an inverse correlation. Where neutrophils are, there's low hypoxia, but then surrounding them, right, in these regions right here, you have high pimonidazole staining, which to us, combined with the human data, that more hypoxic lesions have more neutrophils, suggests to us that maybe neutrophils are consuming oxygen, and that is triggering this hypoxic environment in Leishmania-infected lesions. So to test this, we infected mice with Leishmania major, and then we took away neutrophils or not, and then asked, does that change how CD8 T cells behave? And what we found is that if we stain for pimonidazole, but now by flow cytometry, CD8 T cells in the absence of neutrophils have significantly less of a hypoxic response. And furthermore, these CD8 T cells also produce significantly less granzyme B lesions. So to summarize this in a cartoon, what we think is happening, and this is not only neutrophils, but we can talk about that discussion, but it seems like when neutrophils, because they're so abundant and recruited early on, when they encounter Leishmania parasites, trying to control the parasites by producing reactive oxygen species, they consume oxygen, so they essentially become these oxygen sinks from the tissue. This produces a hypoxic environment, low oxygen in the lesions, and then when CD8 T cells come from the draining lymph nodes, they experience this low oxygen, and that leads to a more cytolytic program in CD8 T cells. As you could see, if you take away neutrophils, that's not sufficient to completely blunt granzyme B production in CD8 T cells. So we think there might be other cells, more innate cells that can be doing the same thing, and also other manners of oxygen sinks, such as nitric oxide production, could also be participating in this process. But I think neutrophils was really an obvious thing to look at first. So, okay, this is fine, this is nice. Biology, we understand that neutrophils can promote this low oxygen environment in the skin that can be translatable to other inflammatory disorders, infectious diseases, and that can change how the immune cells in that environment behave. But what does that mean then for Leishmania patients? I don't think targeting hypoxia is an ideal way of treating this disease, because hypoxia will do different things to different cells. But I think what this is arguing is if we want to really use CD8 T cells as a target for immunotherapy, for example, we can think that targeting the skin itself, it would be sufficient to control disease. We don't need to give a systemic immunotherapy that could lead to, other issues, systemic issues for the patient. We could think about topical treatment for blocking CD8 T cell function. So we decided to test that. So for this, we used a different model of rat knockout mice that when we give CD8 T cells alone, these mice have severe disease. So this is the worst outcome that we can have, which is a good way of treating, testing a drug. So if we infect those mice, wild-type CD8 T cells, we induce a lot of pathology. So what we decided to do was to use a perforin inhibitor that was developed by a group in Australia that's had a systemic version of this drug, and they've developed a topical version of this inhibitor for us, so we could then wait for the lesions to develop and start treating mice. And we started treating every 12 hours to see what happens to the lesions of these mice. Is topical treatment sufficient, and is this really localized to the skin? And what we found is that when we use this topical perforin inhibitor, you control disease caused by CD8 T cells. You have less pathology, meaning less ulceration, loss of tissue. But one of the things that we already knew is that this shouldn't affect parasite controls. So the number of parasites is the same, which argues that you need antiparasitic drugs in combination with these therapies in order to achieve the best outcome for patients. So to summarize what I've told you today and then put some open questions here, instead of beginning here, we think that this model really begins when neutrophils are early recruited within the first few hours of infection and then encounter the parasites, and they produce this hypoxic environment. And essentially, you have this feed-forward loop that leads to more severe pathology. So there are many open questions and the things that we're now interested in studying in the lab. I'll point out one that's related to vaccine development, right? So is this a pathway that we should avoid when we're thinking of vaccine development? Is this in any way impacting secondary responses to infections? So this is an open question and something that we're studying right now in the lab. These are the people in my lab. I have to thank Erin again for doing all the work. Particularly, I'd like to thank Joe Trapani in Australia that has developed the perforin inhibitor, the topical version for us to test, the funding sources, and I'm happy to take any questions.
It seems like we've lost Blakely.
Maybe option. Oh, we lost Blakely.
I'm here. Sorry.
Oh, hello.
I'm not going to turn my camera on because I'm having some connectivity issues here, but I can see.
Okay.
So thank you very much for the talk. If everybody could hold their questions until the end, we're going to do a joint Q&A, and we're going to hand over now to Rachel, who's going to talk about Buruli ulcer and Mycolactone. Thank you.
Thanks, Blakely. Wonderful talk, Fernanda, and hopefully you might see some overlaps actually between the two by a very different mechanism. And I would have to say that actually there's a big thunderstorm passing over here at the moment. So I'm very much hoping that that it's not going to interrupt my my connection. So hopefully everyone can see the right screen here.
If we lose you, we'll assume you got struck by lightning. I think that's the
yeah, it seems possible. It's quite intense actually, but anyway, fingers crossed, everything will be fine. Okay, so yes, yeah, thank you, Fernanda. It was such an interesting talk. I'm going to be talking about a completely different pathogenic mechanism that's in the disease Buruli ulcer that's caused by Mycobacterium ulcerans. Now, this slide shows the scope of the research that we do in my group that spans all the way from medical illustrations done by a wonderful PhD student and ongoing collaborator in my lab, Joe Butler, all the way to working with the purified version of Mycolactone, which you can see on the bottom right. But we work at all the scales in between, from cellular, molecular through tissue and with patients as well. But what I'm going to try and achieve in the next 20 to 30 minutes is try and explain the mechanism of Mycolactone action at Sec61, because it's about a decade now since we discovered this mechanism, and it's really clear to me that it's quite complicated for people to get their heads around, being a bit of cell biology that people perhaps haven't thought about since they were at university, or even if they studied it at university. So I'm going to try and explain that mechanism of inhibition, and then I'm going to describe in detail how it impacts two areas that are relevant to vaccines, and that's around vascular dysfunction and immunosuppression. So let's dive straight in. So I think the best way to explain inhibition of Sec61 by Mycolactone is by analogy. And the best analogy that I was able to come up with a little bit of help from Chat GPT, was thinking about Sec61 translocon as a baggage handler. So in this analogy, we have people traveling from London to Bogota, and they travel via Madrid, as is typical when you're transiting in a long distant journey. And people want to get their luggage from London to Bogota, and that requires some way of Madrid airport directing baggages in the right direction. So here we have people's baggage in London. About a third of that luggage is going to be going to Bogota, but the rest of it is going to stay in Madrid because people are going on holiday there. So the first step of the journey is quite straightforward, but in Madrid, that baggage has to be sorted. So in our analogy, these luggage are proteins. And so only selected bags or proteins are allowed out of the airport, which in this analogy is the cell, and only if it has the right luggage label, which in this case is the signal peptide. So if our luggage has the right label, it can go to Bogota. If it doesn't have a signal peptide, it remains in Madrid. So this is fine. This is how we expect things to work. But if this doesn't work, then we've got two problems. The first problem is that those people going to Bogota won't get their luggage because it's not being directed to the same place. The second problem is that Madrid airport ends up looking like this. Now, we think this is the main way that cells die in response to mycolactone. You get all this accumulated protein that isn't in the right location within the cell. Okay, so let's get a little bit more sciencey now and go through this analogy in terms of cells and proteins and secretion. So here, this is our transit airport, which is the cell. The luggage is proteins. And the ones that are coloured need to go through the secretory pathway to their destination. And the white ones are going to stay either within the cytosol or then be targeted to the nucleus or other organelles like the mitochondria. And it's the signal peptide which targets proteins to the secretory pathway. And that very often takes place through this protein called Sec61, which I've illustrated as a donut because that's very much the structure of the protein. So secretory proteins use Sec61, as do membrane proteins that end up in the cell surface, or other sorts of proteins that end up in the secretory pathway, like in the Golgi. Okay, let's get a little bit more sciencey again. So here we've got a nice cartoon of the Sec61 translocation and the translocon and the process of translocation. So what happens in practice is when a ribosome starts to translate a protein with a signal peptide, the ribosome ends up being attached via receptors to the Sec61 translocon. Translation is actually paused until this binding takes place, and then translation takes place and actually then directly translates those proteins into the ER lumen. That's true of all the proteins containing a signal peptide. There's another group of proteins containing a signal anchor, which are transmembrane proteins, and there the proteins are inserted via their signal anchor or transmembrane domain into the ER membrane. And you can see there's quite a wide range of different types of proteins that use this mechanism. And in fact, it's about 30% of the proteome, so a significant proportion. And very important pathway, and not one that that we think about very often unless we end up working on it. So can I get this to work? Right, okay, so here we have an illustration of mycolactone. You can see where it gets its name from. So myco, because it's from a mycobacteria, and lactone, because this chemical compound is a polyketide lactone. So it has this 12-membered lactone core and these two lipid-like side chains. So this molecule is very hydrophobic and lipid-like. Thanks to really groundbreaking work by Pamela Small's group back in the 1990s, we know that this chemical is responsible for the pathogenesis of Buruli ulcer and can itself cause ulcer formation. So Buruli ulcer is a skin lesion which is painless, and we know that mycolactone is responsible for that as well, but I won't be talking about that today. I don't know why I can't move these slides. Right, here we go. So, so in terms of what mycolactone does, it has these it was known for a long time that it had these very broad pleiotropic effects, and they fall into three main categories. So the first category here shown on the left, we call cytopathic and cytotoxic, and they include the fact that it induces apoptosis, cell cycle arrest, cytoskeletal rearrangements, loss of adhesion, and induction of various stress responses, including the integrated stress response and selective autophagy, perhaps because it induces calcium leak from the ER. And then it has a whole bunch of effects which mean that it's immunosuppressive. So it's known that it inhibits T cell function, including the production of T cell cytokines, it inhibits antigen presentation, co-stimulation by dendritic cells, as well as the innate immune response by inhibiting the production of inflammatory mediators and cytokines by monocytes, macrophages, and so forth. And thirdly, it has these effects causing vascular dysfunction, which include increased vascular permeability, loss of coagulation control, and loss of the basement membrane. Interestingly, quite recently, it was shown that mycolactone is also pro-inflammatory, so it induces IL-1 beta via the inflammasome, which is interesting in the context of its broadly immunosuppressive effect. So in the past 11 years now, a lot of these have been shown to be Sex61 dependent, and they're indicated here in yellow. We've done a lot of that work in my group, indicated here, but I just really wanted to highlight that this mechanism has been investigated by a lot of different labs, including Caroline Demangel's lab, who's played a really important role on the immunosuppressive side. It's really clear now that this is the major pathogenic target of mycolactone, which was quite exciting because for around 20 years or so, it wasn't known how mycolactone caused the pathogenesis seen in BU. Okay, and so here's some of the dates for some of those findings. Okay, so we've had to work quite hard to convince people that this is the major target, and I'm just going to show some snippets of work that we've done showing that Sex61 is the target of mycolactone. And quite a long time ago now, in 2020, working together with Matt Higgins and Sam Gerard at the University of Oxford, we solved the cryo-EM structure of mycolactone bound to Sex61. So in this structure, you can see the structure of Sex61 here. It's a molecule that has a gate that can open and close between some of its transmembrane helices, and you can see mycolactone here in pink actually wedging in between those structures. And here's the PDB file where you can see Sex61 within the ER membrane and mycolactone nestled in there in a way that prevents the signal peptide from accessing Sex61. And that work also involved Belinda Hall, who's a senior postdoc in my lab. But Joy Becci, who did her PhD in my lab, had also done some work showing that we can generate a variety of different mutants of Sex61 alpha, this major subunit of Sex61, that render cells insensitive to the effects of mycolactone, so they can survive and divide in the presence of mycolactone, showing that this is really the cause of cytotoxicity in the cells. We've also done quite a lot of work looking at what sorts of Sex61 substrates are affected by mycolactone. We did this in collaboration with Steve High, Mike McKenna at the University of Manchester. So here we have our the same list of the types of proteins that are affected by mycolactone. Just to remind you again, a bit of undergraduate biochemistry. These proteins can have different topologies. We can have secretory proteins with signal peptides, which is removed during straight after the translocation process. We have type 1 proteins where the N-terminus is outside the cell, where the signal peptide is also cleaved. We have type 2 proteins, which have the opposite topology, and these use the signal anchor and don't have a signal peptide. And then we have a range of different sorts of polytopic proteins, which span the membrane multiple times that may or may not have a signal peptide. And this little table summarizes quite a lot of work showing that mycolactone inhibits all secretory proteins and almost all type 1 and type 2 transmembrane proteins, with a slightly more mixed picture for polytopic proteins, where the presence of the signal peptide seems to be pretty important. In the last 5 years, there's actually been some quite significant advances in our understanding of protein translocation. And to summarize all that, the take home really is that where any substrate depends on Sec61 to be translocated, then it is inhibited by mycolactone. If it uses another translocon, like the GET pathway or the EMC, then it can escape mycolactone's effects. So we think this is kind of explaining why we see these pleiotropic effects. But the interesting thing in terms of trying to understand how to help people with Buruli ulcer is exactly how it's affecting the host response, so that maybe we can support the host during antibiotic treatment. And also it means, because this effect is post-transcriptional, unfortunately, we can't do transcriptional analysis because we know that we do see transcription of genes which are then no longer made because of mycolactone's effect. So we have to use protein approaches. And so a few years ago now, Belinda did some quantitative proteomics on primary endothelial cells. And we enriched for the membrane proteins in membranes and did quantitative proteomics using TMT. We're very happy to identify more than 6,500 proteins. And you can see that, that this data in Cellular is in line with the in vitro data, showing that the type 1 and the type 2 proteins, as well as the secretory proteins, are very much affected by mycolactone, whereas the multipass proteins, it's only a smaller proportion, and that's mostly those that contain a signal peptide. So the type 3 and tail anchor proteins here use these alternative translocons and aren't affected by mycolactone. And then if we look at the cellular compartment of these proteins, you can see that the most affected is the Golgi in terms of the proportion of total proteins that are downregulated, and followed by the lysosome and the ER and the ERGIC, which is the ER-Golgi intermediate compartment. So we think this is having pretty profound effects on all cells that are exposed to mycolactone. And because mycolactone diffuses away from the bacteria within tissue, this is affecting all the cells that kind of are around where you get these clusters of acid-fast bacilli in the disease. Okay, so in the second half of the talk, I'm just going to not discuss these cytopathic cytotoxic effects at all. Very happy to answer questions about them, and I could probably talk about them for another hour. Instead, because they're more relevant to the vaccine side of things, I'm going to be talking about the vasculature effects and the immunosuppression, because both of these, I think, are a bit of a challenge in terms of developing a vaccine. So let's talk about the vasculature first. So the vasculature obviously consists of large and small vessels as well as capillaries, but all of these contain this endothelial layer, which is a single layer of cells, either making up the capillary or in the contact point between the blood and the blood vessel. And they are enwrapped by parasites and then go within the vessel structure itself. And then the inside of the endothelial cells has this structure called the glycocalyx, which is this heavily glycosylated proteoglycans, which are is quite now known to be quite a dynamic surface that's actually regulating the immune system and blood coagulation within vessels. And so the types of ways that endothelial cells can change during infection or any sort of illness are shown on the right here. So we have a range of proteins, anticoagulant proteins in particular, that can help regulate blood clotting on the cell surface. And then we have proteins involved in platelet binding, like von Willebrand factor, as well as the ability to respond to nitric oxide. And it's really important to understand that a lot of these processes are controlled by secretory and membrane proteins. So we reason that they're likely to be affected by the Sec61 mechanism. And also interesting is that they're profoundly altered by IL-1 beta signaling, and we now know that that's likely to be an important part of the pathogenesis story in Buruli ulcer because of Estelle Marion's work. And actually, since the mid-20th century, it's been known that fibrin deposition is widespread in Buruli ulcer lesions. So here I'm showing some punch biopsies of Buruli ulcer lesions in collaboration with Gerd Pluschke. And you can see these very typical staining for Buruli ulcer on the left here. So in the H&E stain, you can see the necrotic subcutis and the intact dermis that you see in BU patient skin. And this layer of acid-fast bacilli, which you can see in the blow-up here, which is the source of the mycolactone, which is diffusing through the tissue. And when we stain similar punch biopsies for fibrin, you can see in the brown here that there's really widespread fibrin within BU. And notably, it's in the tissue as much as in the vessels, which is quite unusual and unexpected. And so just to, before we get into the blood clotting side of things, just to remind people about the blood clotting pathway that involves the initiation by prothrombin, which forms thrombin, and then thrombin can go on to cleave fibrinogen, which is present in large amounts in plasma, into fibrin, which creates this mesh, collecting the platelets and forming the blood clot. But fibrinogen should be in plasma, it shouldn't be in tissue. It's a high molecular weight protein that's around, that should be contained within plasma. Okay, so if we start to think about these proteins in our proteome, these are secretory and type 1 transmembrane proteins. This work was done not only by Joy, but also by Louise Sear, who is a postdoc in my lab. And am I going to be able to do this? There we go. And so what Belinda and Joy did was they, using thrombomodulin as a model and linking it with GFP, we did a time course of staining of thrombomodulin expression on non-endothelial cells. And here we can see that the thrombomodulin depletes over the course of the experiment. I hope you can see as well, we're seeing all sorts of interesting things. We're seeing cells attach and reattach, which is quite important. And if we look at a quantification of this, which is shown in the dotted black line, you can see that this is consistent with the loss of thrombomodulin at a rate of a T-half of 8 hours. And actually, this is the known half-life of thrombomodulin. So that means what's happening here is that the thrombomodulin is being lost as it naturally turns over, but it can't be replaced because of its inability to be translocated. And then here on the bottom left here, we've just got some IF staining showing that von Willebrand factor is also lost from these cells. If we then think about adhesion molecules in the basement membrane, I mean, there's obviously a huge number of these, and we detected a lot of them in our proteome. And this heat map here is showing the extent to which protein abundance is increased or mostly decreased. And you can see that we've got a widespread decrease in these adhesion molecules and basement membrane proteins. And just to pick out an example here where we validated laminin alpha 4, which you can see is almost completely lost from primary endothelial cells within 24 hours, as is most of, but not all of the integrins. And then for proteoglycans, which make up the glycocalyx, there's quite a few of these, and they have different sorts of modifications of them, including heparan sulfate, chondroitin sulfate, and dermatan sulfate. And you can see that we lose this heparan sulfate from primary endothelial cells as well. In the wild type cells, you have this beautiful network of glycosylated proteins, which is lost after 24 hours with mycolactone. And why is this? Well, I know this is a very busy slide, but what I want to what I want you to take away from it is that all of these proteins in capital letters are proteins of the glycosylation pathway that are making these second order glycosylations, which we know are important for the glycocalyx, and all of those in red are significantly reduced by mycolactone. And I hope you can just take from that that there's a huge amount of red here. We're losing almost all of these enzymes, perhaps not surprising because most of them are type 2 transmembrane proteins. And what we did is we validated B3-GALT6, which is the third step here, which should be normally present in the Golgi. And hopefully you can see after 6 and 12 hours of mycolactone treatment that the amount of B3-GALT6 is decreased quite a lot, which I think is probably closest in this blow up here, which means that the cells can't undertake any of this glycosylation. And last but not least, we have this category of proteins called junctional proteins that help the endothelial cells bind together and make this tight junction so that the vessels don't leak, and we lose them. And again, if we validate these by Western blot, we can see that we lose the ties and JAM-C. And the effect is much more profound than it is for these known mediators, which are known to increase vascular permeability, like LPS and IL-1 beta. So again, this work was done by Louise, and we were interested in how this affected blood vessel integrity. And so we did some experiments where we looked at the diffusion of FITC dextran, and showed that mycolactone is almost as effective as IL-1 at increasing this permeability. And actually, when we look in patient samples, we can see this following itself out. So in this example here, we're showing tissue factor staining at the basement of membranes. But if we look in Buruli ulcer patients, what we see is not only is this tissue factor no longer contained within this basement membrane layer, we see fibrin forming very close to those vessels. We've also fairly recently established the mouse model of Buruli ulcer in our high containment labs here at Surrey. And here I'm showing mouse foot pads that have been infected with M. ulcerans. And what's really interesting about this is that in in the later stage lesion, we see, like in human patients, this fibrin all widely distributed within tissue. In the earlier stage lesions, we can see fibrinogen starting to penetrate the outside of those vessels, which is most unusual. And we think that's the stage at which we're starting to get some leakiness. And actually, if we do electron microscopy on these early stage lesions, which was done by Sonia Santana Varela in my lab, you can see that we start to see this breakdown of the basement membrane, I'll turn my laser pointer on again, that you can see that these all of this breakdown of this basement membrane structures here. And a big thank you to Javier Salguero and Jose Diaz Delgado, two pathologists who help us understand these immunohistochemistry images. Okay, so I've got a few more minutes. I'm just going to talk about the anti-inflammatory side of things. And this is where I really started researching this when I was a postdoc, looking at cytokines produced by monocytes and macrophages. And this was the conundrum that really started my interest in the mechanism, because I think it's, as you can see in these dose response experiments showing that mycolactone dose dependently inhibits the production of TNF, IL-6 and COX-2 in these cells. The mRNA levels in these same cells are the same or even increased, which is kind of confusing because normally you would expect them to correlate quite well. But of course, this is quite nicely explained by the Sec61 inhibition mechanism. And in fact, a similar thing is shown in T cells. So in this example from Caroline's lab is looking at interferon gamma, which is inhibited by mycolactone, but the mRNA levels are the same or higher in those same cells. So the mechanism is not cell dependent. And if we do a wider array type approach, where we're comparing cytokines that are made by monocytes in the presence, macrophages in the presence of LPS, and the presence of LPS and mycolactone together, I think you can see that we're seeing this broad span loss of cytokine production. And where do the cytokines go? Well, I still like to show this because Belinda did this experiment. This is quite a long time ago now, and still one of the most exciting results I ever saw, because I'd been trying to understand how we could lose these mediators in the presence of mycolactone. And when we undid the proteasome inhibitor, we could actually restore those proteins. So if you compare this box here, sorry, the big thunder, where we can't see TNF transiting through cells, in the presence of the proteasome inhibitor, we can see it reappearing. And in fact, that material is in the cytosol of cells rather than in the membranes where it should be, because of the translocation mechanism. I think I'm a bit short of time, so I don't know if I'm going to talk about IL-1. I could maybe talk about it very briefly. So
got a couple of minutes. Go for it.
Thanks, Blakely. So this is really very interesting work from Estelle Marion's group, which is something that we'd sort of been thinking about as well, because of course here we have our sort of model for what happens for conventional cytokines. So what happens is you get an inflammation, you get transcription of your mediators, and those transcripts get translated, and those proteins go through the secretory pathway. But when mycolactone's there, they are inhibited, and so we know now that that's why that we don't see those cytokines made. However, IL-1 beta is a completely different story. So instead of being transcribed and then being co-translationally translocated into the ER system, pro-IL-1 beta is made in the cytosol. And then thanks to this really amazing mechanisms that I think I still don't understand fully, you get, in response to what's called signal 2, you get the assembly of these enormous inflammasome structures, which contains multiple proteins and multiple copies of those proteins. And when this structure assembles, you're able to get activation of caspase 1, and then caspase goes on to cleave that pro-IL-1 beta into its active form. And actually it also does the same thing to another interesting protein called gasdermin, which when it's activated can go on to form pores in the outside of cells. And those pores are thought to be one of the main ways that IL-1 beta escapes the cells in a process called pyroptosis, which kills cells. There are other ways that IL-1 can get out of the cell via secretory lysosomes and via this interesting pathway involving secretory autophagosomes. But the interesting thing about it is that none of these steps involve Sec61 alpha. And so my original thinking was that IL-1 beta wouldn't be as affected as the other cytokines by mycolactone. But what Melanie Foulon showed was that, in fact, mycolactone can act as signal 2. So in this bar chart on the left, if we compare LPS that's on its own, or let me just see if I can remember what it is. So LPS is on its own, is on the left-hand bar. And then we've got this middle high bar here. This is a positive control inducer of the inflammasome, of signal 2. But we can see that mycolactone also induces signal 2, so increases the production of IL-1 beta. And then just to talk very quickly about the way that mycolactone affects adaptive immunity. So this is work that's been done almost exclusively in Caroline's lab. But she's shown that mycolactone inhibits the production of both class 1 and class 2 MHC, as well as CD3 and CD86. And so obviously, you know, if you can't make CD86, you can't have co-stimulation of T cells. And then a really interesting phenotype in T cells where if you add mycolactone and look immediately afterwards, you don't see any difference in T cell function. But if you add mycolactone for 24 hours and then try to get a T cell response, you see a significant reduction. And this is likely due to the loss of all of the ability to make cytokines and all of those processes that involve the secreted in membrane proteins. Last but not least, I wanted to talk about some work which we're just looking for a home for at the moment about the loss of lysosome function. And this work has really been led by Belinda, who's a really fantastic cell biologist. And so this really interesting molecule here is the VATPase. It's absolutely essential for lysosome function, and it contains lots and lots of different proteins to make up this super complex. Almost all of them don't have a signal peptide, and so aren't mycolactone targets. But there are a couple of them, ATP6AP1 and ATP6AP2, which are type 1 transmembrane proteins, and they were reduced in our proteome. So we asked whether lysosome function can be affected by mycolactone. And in fact, we can validate the loss of these proteins in cells. And we can actually track the loss of lysosome function, although it does take time for this phenotype to emerge. So in this top illustrating here, we're looking at a marker called DQ-RED BSA, which is only fluorescent when it becomes cleaved away from its quencher within lysosomes. So in normal healthy cells, we see a lot of purple staining. When we add an inhibitor of the VATPase BAFF-A1, you can see we see a lot less pink. But in the presence of mycolactone, we see this time-dependent loss in that function, ability of lysosome to chew up that BSA. And then if we look at the acidity of those lysosomes, we can use another interesting dye called acridine orange, which is green normally, but only goes purple when it's in an acidic environment. And you can see we see a similar pattern there. And so I'm going to skip over that and summarize by saying I hope I've really hammered home to you that this ability of mycolactone to inhibit Sec61 really is affecting so many different pathways in the cells. And so perhaps it's not such a huge surprise that it is such an important virulence factor. And perhaps it has taken over from all the other virulence factors. And it's interesting to note on that front that the M. ulcerans bacteria has actually lost quite a lot of genes in its genome in terms of pseudogenization and so forth. And that's something we're quite interested in looking at the moment. So thank you, Blakely, for letting me waffle on. And I will just finish, if I can see this, by thanking everyone who's worked in my group and all my collaborators near and far, and leave you with this very cool structure of mycolactone bound to Sec61.
Thank you so much, Rachel, and thank you, Fernanda. So we're going to go to the Q&A now. As I said, the best way to do this is if you can click on the reaction button and raise your hand, which is like this. Can't actually do it myself. If you click on, if you have a question to ask, we have one. Fernanda has a question. Fernanda, I guess you have a question for Rachel rather than yourself. So go ahead.
Yeah, I think Tuhina was first. I think you shouldn't use the reaction button, but I will go because I'm unmuted. So I was going to ask you, and maybe you said this and I missed. So is that a unique thing to Buruli? Like, is there other pathogens, other organisms that have something similar to mycolactone? Maybe you said and missed and I missed
No, I
this,
didn't.
or-
I mean, there's a whole, I mean, it's one of those really interesting things. You have to be a bit selective about what you talk about. So
Yeah.
amongst the mycobacteria, and human pathogens, actually, Mycolactone is unique. So the other, like leprosy, tuberculosis, don't make anything like Mycolactone. They do make a lot of these polyketide type molecules, but nothing like Mycolactone. But there's a small handful of Sec61 inhibitors known in science, and they're all natural products. So they come from marine sponges, from other bacteria, from fungi. So there does seem to be some convergent evolution, but in terms of bacterial or human pathogens, it's the only one we know of.
Okay. That's very cool, by the way.
So if you have a question, raise your hand or maybe just type in the chat. And we have Tahina. Go ahead, Tahina. Turn on your camera, say hello.
Thank you, both Dr. Fernanda and Dr. Rachel. Awesome talks. And I'm just surprised at looking at how these various microorganisms are also very similar to Mycobacterium TB, how the pathology looks very similar. And I do have one question for Dr. Rachel. And that is, have you looked at secretion of matrix metalloproteases or MMPs in presence, like does Mycolactone promote MMP production from these cell types? Like, especially when you're looking at the vascular leakage.
Thanks, Tahina. We haven't specifically looked at them in terms of validating our data set, but most of the matrix metalloproteases are either secretory proteins or some of them are GPI anchored, I think, aren't they? So we would expect them to be lost. So you might see an induction of expression at the transcriptional level, but because the cell can't make them, so the idea is that so the reason why perhaps I didn't explain this very well, but the reason why we don't see the proteins when translocation is inhibited is that those proteins are translated in the cytosol instead of into the ER membrane. So they don't enter the secretory pathway. And so they are degraded by the proteasome, or some of them are removed by autophagy. But eventually they aggregate within the cytosol of the cell. But we haven't looked at matrix metalloproteases. It would be interesting to look through our data set and check for them. I know they're very important in TB, aren't they?
And is there, I know this is a very naive question, but is there a way to scrub Mycolactone
from tissue?
Yeah.
Yeah, I mean, well, so this is another really interesting question. We don't actually have a way of measuring Mycolactone in tissue as yet. We don't have the we don't have the luck of working with a protein that we could tag in some way and then track in our experiments. Lipids are very tricky to work with. The way that one deals with tissue normally is you embed it in wax and then you wash it with solvents, which we think would remove the Mycolactone. So it's not known exactly where the Mycolactone is. And some people, so Richard Phillips' group in particular in Ghana, have tried to quantify Mycolactone in lesions during antibiotic treatment. And there's differing evidence. Some evidence shows that it sticks around for a long time. Other evidence says that it disappears quite quickly.
Thank you. And I have one more question, if I'm allowed, Blakely. Okay. For Dr. Fernanda, so the Leishmania, do you see various virulent, what do you call it, subtypes? Like clinical isolates, sorry, wrong.
So there are clinical isolates that are more severe than others. There are people that are studying. I don't work with, it's funny because I've been working on Leishmaniasis for 20 years and I'm now just starting actually working with the parasite. But there are people looking into virulence factors and things that are maybe different between some isolates, particularly for Leishmaniasis, and seeing how they respond to different drugs and other things. Yeah, so there's
Yeah,
some that are cause more severe disease than others.
yeah. The reason I was asking is because we see the same thing in MTB, where if a MTB is more virulent, you will see more neutrophils coming into the area and more hypoxic granulomas. So that's the whole reason. But thank you.
Thank you. Blake, you're muted, but Rachel, I guess you can ask.
Okay, yeah. Yeah, so Fernanda, I enjoyed your talk so much, and I was really struck in particular about the importance of hypoxia in visceral leishmania, and you can see how I'm seeing these overlaps with fibrin deposition, which I think is quite important in Buruli ulcer. But my question is whether you think that this mechanism is a pathological mechanism that the parasite is trying to induce, or whether it's just a byproduct of the host immune response. Because you saw differences in pathology, but no difference in parasite number. So is it helping the parasite, or is it helping the host?
So I'm not sure. I think about this a lot, whether there's some advantage to the parasite in all of this. And that's why actually I am trying to work with the parasite now after such a long time. I want to understand whether we don't know how Leishmania adapts to a hypoxic environment. We can put a macrophage infected with full of parasites. The parasites are okay in an infected cell that is under low oxygen. So somehow the parasites can adapt to this. We don't know the mechanisms that Leishmania adapts to low oxygen levels. It doesn't have hypoxia-inducible factor homologs like mammalians do. So we are trying to understand how they adapt. So it's hard to say if it's a cause and consequence and the parasites just benefits from it. My inclination is to say that this is an adaptive mechanism that has evolved for the host to adapt to tumors, for example, right? So in tumors, they're very hypoxic, which is more prevalent than Leishmaniasis, and arguably would be a greater pressure, right, for the host than a Leishmania infection. So tumors are highly hypoxic. So I think this cytolytic response is an adaptation of CD8 T cells to tumors, right? But then in Leishmaniasis, for some reason, this outcome, which is positive in cancer, it would be positive in a viral infection, for example. We kill the virally infected cell, the virus is gone. But for Leishmania, this somehow became beneficial. So whether the parasite induces this as a mechanism to survive, I'm not sure, or if this is just a byproduct of, something that should be positive and ends up being negative for Leishmania. But it's a hard thing to answer. I'm trying, but it's difficult. But thank you for the question.
That's really great.
Blakely, you're muted.
Sorry, I muted myself. Veronica, go ahead.
Hi, good morning or good evening. I don't know. For me, it's good morning. And nice talk, Rachel and Fernanda. Nice to meet you. I have a question for Fernanda. If could boosting local oxygen help heal Leishmania lesions, or the early neutrophil which make unusable, this kind of strategy? Because I remember that I read a paper a long time ago that in Crohn's disease model that the migration of neutrophils to the inflammatory sites causes the depletion of the oxygen. I know that there is a change in the inflammatory profile of the cells, and I'd like to know if it's a possibility, because I know that there are some oxygen therapy for skin lesions.
Yeah, so there's a lot of interest in dampening hypoxia, right, because of tumors, because it has this positive impact on solid cells and solid tumors. So there's a lot of interest in inhibitors for HIFs and things like that. I would stay away from that necessarily. It's not something I would try to target, because There's so many different effects. HIV can have different effects on CD4 T cells and neutrophils and macrophages that it's hard to, for Leishmaniasis at least, because it's an infectious disease, I would be careful with that because of this. However, there is data in the past that people used to treat Leishmania lesions with heat. And one of the mechanisms they think that it works is increased vascularization of the tissue. So maybe indirectly by increasing vascularization, you can actually dampen the hypoxic microenvironment. That could have a positive effect. But I think there's so many other strategies that are easier and also more feasible with drugs and immunotherapies that target these pathogenic responses that I think that I don't think something like this would be necessary. And it's more difficult also to implement, I think.
Okay then. Do we have any last minute questions? I'm going to give you 20 seconds to have a think of a question. If not, I think we're going to thank our two amazing speakers. Okay. Thank you so much, Fernanda. Thank you so much, Rachel. Two excellent talks and some really excellent questions there. If you could all in the chat now somehow express your appreciation. We usually just do it through React using the React button. But yes, I just want to say thank you very much. And if I'm sure if you have any further questions, you can either email the validator or we'll pass them on, or you can get in touch directly with the our two speakers. You can find their details on the Validate website. Well, thank you very much.
Well, thank you, everyone.